All right, welcome back. This is going to be part 4 in our excitable cells lecture. And so here we're going to discuss something known as the Nernst equilibrium potential, or the Nernst equation. And so what this is saying is that the equilibrium potential, and we'll discuss what an equilibrium potential is in a second, but the equilibrium potential for a particular ion, so sodium or potassium, is going to have an influence on the membrane potential. All right, and so it was initially thought when this idea of an equilibrium potential was first being discussed that the membrane potential was equivalent in the same thing as the potassium equilibrium potential, and that was very close to being true. But it turns out that as the equilibrium potential for potassium changes, and again, we'll discuss what equilibrium potential is in a second. But as the equilibrium potential for potassium changes, so does the membrane potential, the resting membrane potential, in the same direction, but then it starts to get off a little bit. So it doesn't follow the potassium equilibrium potential exactly, but it does follow it very closely. All right. So what is an equilibrium potential? So an equilibrium potential for a particular ion is the balance of two different forces. All right, you have an electrical force, so that's the electro part of the electrochemical equilibrium here, and then you have a chemical force, right? So the electrical force is the charge force. So in other words, opposites are going to attract, like charges are going to repel each other. As you have the movement of positively charged ions to one direction and accumulate in one area, the accumulation of positive charge in that area is going to repel additional positively charged ions from moving to that area. Okay. Now, so you have the electrical part. You also have the chemical part. The chemical part is the concentration gradient, right? So you have a high concentration of sodium on one side of the membrane. It flows down its concentration gradient towards the lower concentration, right? So in other words, let's take sodium for example. Sodium moves from a high concentration to low concentration outside of the cell to the inside of the cell. So that first force is just the concentration, right? We move from high to low. As you accumulate more and more sodium ions inside the cell, you're accumulating more positive charge inside the cell, right? So the membrane potential becomes more positive. That positive charge now repels the additional movement or the additional positively charged ions, right? So more sodium. So as you accumulate more and more positive charge in one area, that positive charge repels additional movement of more and more positively charged ions, in this case being sodium, all right? So where these two forces come to balance, so where you have the electrical force becoming balanced with the chemical driving force, that's the equilibrium potential, right? So this is where you have no more net movement of an ion one direction to the other. And you have this for both potassium, you have it for sodium, but you also have it for chloride, magnesium, calcium. Every ion has its own equilibrium potential. And an important note, and this is very important for you to understand, is that each ion tries to make the membrane potential closer to its own equilibrium potential. So we'll see that the equilibrium potential for potassium is very, very negative, like negative 90, negative 91. The equilibrium potential for sodium is very positive at around positive 60. And so when sodium and potassium move, respectively, they try to move the membrane potential closer to their own equilibrium potential, right? So hopefully this makes sense. And so there's a video there that you can click on and watch if you have the access to the slides. So you can see here, so we have a potassium here. We have potassium inside, we have potassium outside. Equilibrium potential, this E sub ion, this is the equilibrium potential for each of these individual ones. And again, focus primarily on potassium and on sodium. So potassium is very negative, somewhere around negative 90, negative 89, something like that. Sodium is very positive, positive 61, positive 62. So as these ions flow across the membrane, or as they're made more permeable and begin flowing, they move the membrane potential closer to their own equilibrium potential. So in other words, as sodium begins flowing across the membrane, it makes the membrane potential more positive, So we said that the resting potential was negative 70. It tries to bring it up closer to its own equilibrium potential of positive 61. But as we'll see, it doesn't quite get there. Likewise, whenever potassium is made more permeable, it starts flowing. It takes the membrane potential closer to its own equilibrium potential, so it takes it closer to the negative 89, negative 90. But again, as we'll see, it does not quite get there. Okay, so if you see here, this is a graphical representation of what I was describing earlier, and how the movement of potassium, or the permeability of potassium, influences the membrane potential of the neuron, but not exactly, right? So you see that you have a extracellular potassium measurement here, and so the more extracellular potassium you have a change in the membrane potential. And you see that the blue and the purple line here are exactly the same, or pretty close to being the same, all the way down until you get to this point around here where the extracellular potassium is somewhere in between 1 and 5. And you get this kind of divergence. So you get this area where the two split apart. So this says that a change in the potassium equilibrium potential does not cause the membrane potential to change in the same manner or to the same degree. All right. So essentially all this is saying is that there has to be some other ion at play. Right. If the membrane potential of the neuron is not exactly the same as potassium, then that means there's got to be some other ion that's exerting an effect. Right. And it turns out that's exactly the case, and that other ion is mainly sodium. All right. So these two things together, these contribute to our resting membrane potential. All right. So this membrane potential of sodium equilibrium potential and potassium equilibrium potential and these movements, you can think of it as though you're filling a bucket of water, right? You're filling a bucket of water from one direction, and at the same time you're releasing water in the other direction, okay? So as long as the flow of water in is the same as the flow of water out, so the influx is equal to the efflux, then you don't really get any major change, right? You don't get a net change in the amount of water in the bucket, right? Because what you're adding is the same as what's leaving. So this is the same principle holds true for our membrane potential. The flow of ions, positive and negative, across the membrane, as long as those are equal going in and out of the cell, then the membrane potential is not going to really change a whole lot. It only changes when you have a major change in either the entry or exit of a particular ion, right? So if we look at this bucket of water, say these flow rates are the same. Influx is equal to efflux, so there's a net flux of 0, so the water level doesn't change. Say you now close off this valve, but you keep adding water here. Well, obviously this bucket of water is going to fill up because the influx, water coming in, is much greater than the efflux, in this case, there's no efflux, right? We turn the valve so the water is no longer flowing out. Likewise, similarly, if we stop the influx of water, but we kept this valve open so water was flowing out, we would see the water level drop severely, right? And again, because the rate of influx and efflux are not the same, okay? So this happens also to our membrane potential whenever we alter the permeability of sodium and potassium. As you alter the permeability of sodium, you increase it. It's going to then flow down its concentration gradient. So where is it going to flow? It's going to flow into the cell. And it flows in at a greater rate than anything flowing out. And so you get a net influx of positive charge. So we see the membrane potential shoot up. We see it become very, very positive. And then similarly, whenever we increase potassium permeability, potassium leaves the cell. It flows out. And when it flows out, it takes with it its positive charge, and that causes the membrane potential to drop back down. Okay, so this is a very kind of easy analogy to remember, and help you kind of understand the movement of ions across the membrane. All right, so when it comes to this equilibrium potential, this is going to be a very important driving force for determining what the membrane potential is. Right, so there's a relationship between the resting membrane potential and the equilibrium potentials of our different ions, namely sodium and potassium. All right. So if we look at this graph, all this is saying, or this slide, all it's saying is that the permeability of an ion will influence the resting membrane potential of the neuron. Okay. As the permeability of an ion increases, the flow of that ion increases, and so the membrane potential becomes closer to, not exactly, but closer to that ion's equilibrium potential. All right. Likewise, whenever the permeability of an ion decreases, the membrane potential goes away from that ion's equilibrium potential. Right. So if we look at this graph, we know that we start at the resting membrane potential, which we said was about -70. Right. So we're starting right here. We're starting at our -70, and say we start here with the permeability of an ion increasing, and say we increase it to we increase sodium permeability first, right? So we open up our sodium channels. Sodium is going to begin flowing. Which direction will sodium flow? Sodium flows from high concentration to low concentration, so it flows from outside to inside. Well, we said that the inside of the cell was originally negative, right? -70, and as sodium flows in, it moves the membrane potential closer to its own equilibrium potential, right? So on this graph, are we going to go left or are we going to go right? Well, if we're moving it closer to the sodium equilibrium potential, you should say we're going to go to the right. So we begin -70, and we begin moving and moving and moving this way until we get somewhere around here, maybe +30 or so, all right? So if the sodium permeability was increased, and we're moving toward the sodium equilibrium potential, why do we stop at about 30 or so, and why do we not make it all the way to +61, right? So maybe pause the lecture, try to answer that, and we'll move on. So the reason it doesn't reach the sodium equilibrium potential of +61, 62 is that there's other ions at play, right? You have chloride, you have potassium, and at the same time, they're exerting their own force on the membrane. They have their own equilibrium potential, which in this case happens to be very, very negative, right? So we don't reach any ion equilibrium potential, but you approach it, all right? So say we're at +30, and then we close our sodium channels, so sodium permeability decreases, and then we open up potassium channels, right? So say now we open up potassium channels, potassium permeability now increases. Potassium flows from high concentration to low concentration and moves from inside the cell to outside of the cell. Sodium is positive. As it moves out, it takes with it its positive charge. And what is the equilibrium potential for potassium? Well, equilibrium potential for potassium is somewhere around -89, -90. And so if we were at +30 and we opened up potassium channels, potassium begins flowing, high to low concentration, out of the cell, and so we begin moving now from right to left. So we start going back this way, and we go past -66, we go to -70, and we go towards -89, but we don't quite reach it, right? And again, why don't we reach -89 millivolts? It's because there's other ions at play that are exerting their own effect on the membrane potential. So sodium and chloride, right? So we end up somewhere around -73, -74, something like that. Okay, but the idea here is that as you alter permeability, you alter the flow of an ion, so its diffusion, and as it flows, it's going to alter the membrane potential to be closer to its own equilibrium potential. Okay, so you can see this same thing here. So this is talking about the resting membrane potential. All right, so we're going to stop here, and we will pick up with our next part talking about some of the structure of neurons, and then we'll get into actually talking about the action potential itself.